good afternoon. You listen me good? Okay. So we will start. And topic of today lecture is teaching of Severtsev, it is famous Russian scientist, on field embryogenesis. And we need little return to our last lecture and speak about human evolution. Human evolution began with primates. Primate development divergent from other mammals about 18.5 million years ago. Among apes, gibbons, orangutans near 8 million years ago. Humans and chimps 7.5 million ago. And in this slide you can see information about stage or period of human evolution. Modern theory speak about or tell about first migration process start from Africa and then disappearance in other areas. This is four stage can describe as the first, which was between 4 and 7 million years ago and consisted proto-hominids. Second stage, around 4 million years ago, and in this period or this stage appearance Australopithecus. The third stage, around 2.7 million years ago, and appearance Paranthropus. And fourth, which was between 2 and 3 million years ago, and appearance Homo sapiens. Modern human appearance about 50,000 years ago, and in last lecture we speak about evolution continues, and our genome, our habitus adaptive to modern environment. If to tell about evidence of evolution, we need to speak about three moments. First, derivative traits are nearly evolved features, such as features that do not appear in the fossils of common ancestors. Ancestral trails are more primitive features, such as teeth and tails, that do appear in ancestral forms. And homological structures are anatomically similar structures inherited from a common ancestor. And in this slide you can see a scheme about evidence of evolution. And we will speak about four directions. Analogs, it is about appearance and development of organs and forms of body. Atavism, and here you can see many hairs in face, tail and other. Rudiments, when for example speak about eyes, you can see some animals has thought eyelid, but human not have, only rudiments inside part of eyes. And homologies, and you can see in picture four different animals arms, or same arm part of body, but in different development. From homologies we start speak about evidence of evolution. What is it? Homologic. It is similar in structure and function because they originated from the same ancestor long ago. If two or more species share a unique physical feature, such as a complex bone structure or a body plan, they may all have inherited this feature from a common ancestor. And to give one classic example, for the forelimbs of horse, human, birds and dogs are pretty different on the outside. And if you speak in picture, you can to note same part of human, dog, birds and hail, which marked as a color: red, brown, yellow, white and dark brown. Some homological structure can be seen only in embryos. For instance, all vertebrate embryos, including humans, have gill slits and a tail during early development. The development patterns of these species become more different later on. Homologous embryonic structures reflect that the development programs of vertebrates and variations on a similar plan that existed in their last common ancestor. And in this at this picture, at this slide, you can see vestigial legs of boa constrictors. It is example of structure includes the tailbone of humans, the hind leg bones of whales, and underdeveloped legs found in some snakes. Next evidence, it is analogical structures. And here we need to speak about many species have similar traits because they are descendants of a single common ancestor. These species developed from a single source and are related to a certain degree despite their current differences. Species may also have similar traits, even though they are not related to each other. This is usually a result because the species live in similar environments and fill similar ecological roles. The structures in this case are known as analogical structure. And this way may be three different ways. Convergent evolution, in this case we will speak about evolution when two unrelated species of a type develop similar structures or genes. One more example. For example, eyes. They present in some species, but development from different starts or begin in different ways, but have same functional and structure. Next way, it is collateral evolution. And here, as example, we can to say about butterflies that belongs to the genus Heliconius, exchange genetic materials by hybridization, mimicry, associated regions of chromosomes. The genes affect the wing patterning. Approximately 30, from 30 till 70% of all flowering plant species have hybridizations, even in their history. One natural hybrid in the sunflowers. Next way or a type of evolution, it is parallel. The development or identical or similar traits in separated species. The result is analogous genes. An example of parallel evolution is monarch butterfly, caterpillar, Danaeus plexippus, red milkweed beetle, Tetraopis tetraphalmus, oleander, aphids, Aphisneria, and large milkweed bug, Onchopeltus fasciatus. The next way of evidence of evolution, it is atavism. Sometimes a trait or a feature that an organism has lost can reappear. Reversals atavism therefore do not produce analogous genes because the gene or genes encoding for the trait has already been present in the organism's genome. Atavism may be caused by the first, a previously suppressed gene become active due to a mutation in a regulatory factors that's suppressed by the expression. And second, a variation of time in fetal development by either prolonging or decreasing the development time. The human embryo initially grows a tail bud and is the most prominent during 32-35 weeks into the pregnancy. But the growth is later switched off. Apoptosis and macrophages will ordinarily remove the tail bud by phagocytosis. However, several reported cases exist where humans also are born with a tail. And in this picture you can see little tail about we speak previously. The kitchen genome contains genes that are involved in tooth development. Also, birds lost their teeth about 60 or 80 million years ago. This is evidence very excellent to show from anatomy or embryology. And Charles Darwin thought of evolution as a descendant with modification, a process in which species change and give rise to new species over many generations. He proposed that the evolutionary history of life forms a branching tree with many levels, in which all species can be traced back to an ancient common ancestor. And in this picture you can see this tree. We will to see this tree evolution of embryogenesis tree one more time, but now you can to see and understand how was development in history from different species and from every species in own evolution. Embryological evidence. For example, in this picture you can see human embryo in right side and chick embryo in left side. Such as tails, postnatal tails, nodes, and gill patches, nodes too. It is the same, but when we will comparative adult chicken and adult human, we not find same. We not find these organs, and we not find morphological or other same between chicken and human. But in this period embryogenesis we can find and comparative as analog or same. And in this slide, in this picture, you can to see more examples and way of development, embryogenesis, about which we speak in last semester, how it happens, many same stages between different animals, fish, salamander, tortoise, chick, rabbit and human. In first stage, many same character. So, embryological evidence more important for understanding evolution, ontogenesis and philembryogenesis. Next way it is organ reduction. In start of lecture I little speak about eyes and explain third eye linked and from birds and absent in eye from human. Here process we can to see in ears, monkey, human and embryo. And next part of this picture, comparative intestine or colon from human and cow. It occurs when any subsystem of the organism loses its functional significance and ceases to be subject to the action of natural selection. It begins to collapse. The first to disappear to those systems that were the latest in ontogeny. Rudimentation organ remained negative anabolism. And example, Astynex jordani cave fish gala. So, all this evidence we need to know that understand what happens in every time in ontogenesis. And this is theory was thankful and leads to scientific word, and then for other one Russian scientific zoologist and evolutionary theorist Alexei Nikolaevich Severtsov. His ideas, such as the theory of philembryogenesis, the concept of morphofunctional and biological progress, remain the basis of research in the field of evolutionary and morphology to this day. So, if you will think why it need to study for doctor, if you will listen careful this lecture, you will understand why it need to study for future doctor. For example, to understand how appearance one or two defect of development. For example, we in last semester we spoke about pathology embryogenesis, about critical period in embryogenesis, and in my group I said about thalidomide effect, when many human child in middle of last century was born without hands or without legs, and group which study in offline regime, we was going to museum, and I know some teachers use video to show our museum exponent for preparation, which show how can development embryogenesis in utero and which problem can be. The second famous scientist which make many for understanding philembryogenesis was Haeckel and Müller. They give to us biogenetic law. The biogenetic law was widely used in the creation of a modern classification of organism or taxonomy, the construction of schemes of phylogenetic relationship of organism. What basis for this biogenetic law? Two very important terms. It is palingenesis and xenogenesis. What means palingenesis? Repetitions or recapitulations of more or less distant stages of phylogenesis in the process of embryonic development of an individual. Okay, what means xenogenesis? According to Haeckel, are signs that violate the manifestations of palingenesis, do not allow tracing the sequence of stages of phylogenesis of their ancestors in the course of ontogenesis of modern forms, that is, they violate the biogenetic law. So we need to understand what means phyllembryogenesis. Evolutionary changes in the stage of ontogeny of an organism occur due to the evolution of subsystems of the organism. Organs, tissue, cells, it became necessary to rethink and transform the biogenetic law proposed by Haeckel and Müller. This was done by a Russian scientist, biologist, and theorist of evolutionary doctrine as a phyllembryogenesis, and this theory was formulated in 1939. In this slide or picture you can see different skulls from Homo habilis, Homo erectus, Homo floresiens and Homo naledi. Here we can to see two types of changes, same which save for all skulls, and different which little nodes in this picture. Next picture again to show development of skulls, feathers and hair of vertebrates. From first to 25 position you can to see, read and understand what happens in evolution with different part and character of embryonic development. And previously I speak about phylogenetic tree or evolutionary tree, and here very excellent pictures which show how happens development and appearance different animals, fish, plants, humans in evolutionary, who and which species more near each other, who very distant in this scheme, who development many stage, for example as primate or horse or as human, and who not development from his start position, for example as protozoans. Try to find in this picture worms, which topic now we studying, and you can to see about little distance from start to the finish of evolution. And if you remember about which morphological structure, which organs, system of organs present in worms, we will understand in real they not need of development because they live in comfort condition and not make any way for development. But it is not real because little, if you will see on branch of these trees, little development evolution was happen. For example, about nematoda and roundworms, we know that at first, for example, Ascaris can to make insight in human organism through skin. When we speak about this topic in our class, we not speak about this type of insight, common insight in organism, because it is only in retrospective evolutionary studying we can to say about this type. But this type was not excellent for this type of worms, and many factors was given damage and more priority became other way through eggs which come to human from mouse, gaster, then in intestine, blood, liver, car, lungs, mouth, and again in intestine. But this is way was or had evolutionary development too, because at first eggs was very weak and not give excellent effect to come inside of human. So eggs cover more strong cover form, or shell, and it is give to this eggs more excellent effect, more successful effect to input in organism. This is a morphological change, neoplasm in embryos that determine new direction of phylogenesis. This is evolutionary changes in the course of ontogenesis. And in Henkel theory, ontogenesis, every organism is a fast and short repetition of embryogenesis. So every human, every animals have in this embryogenesis and ontogenesis. Embryogenesis, it is start stage of ontogenesis, has all stages which this species had in evolutionary process. Only ontogenesis, very shorter and simple, that was in real in this process. In this moment, we make short break time for 10 minutes. Здравствуйте, Юлия Валерьевич. У меня просто была лекция, поэтому я вам не Так, окей, our break time finish, and now we continue speak about lecture, topic of lecture. And in this slide you can to see how happens ontogenesis development in a child, from child before old woman. And what means ontogenesis for memory? Is the individual development of an individual from the moment of the birth, formation of zygote, to the end of life. And next stage we will to say about philembryogenesis, remember, of evolution and ontogenesis. And here we speak about three types of philembryogenesis. Anabolism is an extension of morphogenesis, the addition or additional stages to the development program, the biogenetic law in fulfilled. Deviation is a deviation from the development program as a middle stages of morphogenesis. The biogenetic law is partially fulfilled. And archalaxis, a deviation of ontogenesis. At the very beginning, the biogenetic law is not fulfilled. And next we will speak about every of these type. And the first, anabolia. Further development of the organ from a two-chamber heart to a four-chamber heart. And you can write me who have or which have a human heart, two or four-chamber heart. I think no one student not know about how many chamber had in human organism. But I will continue. At first, all preceding stages of the organ development recapitulate, and only at the end of the embryogenesis mutated genes interfere and a new character of germinated. Next, deviation means diverging from the middle of the organ development course. The development of scales in reptiles. Initially, the process goes according to the phylogenesis, partial recapitulation, and in the middle of morphogenesis mutated genes interfere with the work and the course takes another direction. Deviation in the development of an organ at the middle stages of embryogenesis. An example of deviation in the ontogene of horny scales in reptiles. reptiles. The first stages in this process resemble the onset of development of placoid scales in sharkfish. In this example, it is shown only in early stages of development of scales, and then its development in reptiles deviates from the development of fish scales. Other example of deviation: the formation of horny scales in reptiles, in plants, development of the heart in mammals, and lung formation in mammals. Next, phylogenesis rules. And the first, the rule of the irreversibility of evolution, the impossibility of returning any group of organisms to the state passed before. Second, the rule of progressive specialization. If a group of organisms begin to evolve, then in the future it deepens its specialization in the outlined direction. Third, the rule of origin of new groups of organisms from non-specialized ancestors. And fourth, the rule of adaptive radiation, the branching of the ancestral trunk of a group of organisms in the course of adaptive evolution into separate branches. The philembryogenesis of any group is accompanied by its splitting into a number of smaller trunks which diverge in different directions from the initial level. And Severtsov came to the conclusion that there are the following parts of progress: aeromorphosis or morphophysiological progress, idioadaptation or morphophysiological adaptation, morphophysiological degeneration, and xenogenesis. And here you can see many different pictures which show same organs or derivative of skin from fish, from birds, from human, and from animals. Next, what we will speak it is archalaxis. Are changes from the moment of an organ germination, the development of a hairy integument in mammals. For all that at the beginning of morphogenesis mutated, genes became involved in the work, and the development takes a new course. Recapitulations are almost absent. Changes in the course of ontogenesis can be realized already at the level of phylogeny, aeromorphosis, idioadaptation, and degeneration. Aeromorphosis. There are changes in the structure of the body and in its function which are of general importance and rise the vital energy of organism. Aeromorphoses are very persistent. They are remain unchanged during the further progressive evolution of animals in the form of a permanent trait, retaining their significance in new environment. Aeromorphoses occur only as a result of abrupt changes in the environment or cataclysms. For example, the emergence of living beings from water to land. Aramorphic changes, which led to the progressive development of mammals, were associated with an increase and complication of the structure of the lungs, with the formation of four-chambered heart and with differentiation of teeth. This provided a better way to grip and chew food. The hair cover in mammals was reduced by the change in the temperature of environment. And in this picture, when green step, you can to see that show different type of philembryogenesis. And when we say about plateau, we speak about ideoadaptation. When we see as development direct to increase from one step of development to more high step, it is aramorphosis. For example, parasitic type worms have lost their nervous, muscular, and even digestive system. Nutrients enter their bodies through the integument. These parasites have a highly development ability to produce. Sedentary attached forms may experience reduction, underdevelopment, or a complete disappearance of the nervous and musculoskeletal systems. And other example, bivalve mollusks that switch to passive filtration lost not only the brain but also the head as such. At the same time, their actively swimming relative octopus are called primates of the invertebrate world for their high development of the brain and eyes. The next terms it is ideoadaptations. Private adaptations of the body to a certain way of life in specific environmental conditions. Ideoadaptation does not significantly affect the general level of organization of a given group, in contrast to aramorphosis. Ideoadaptation, there is no improvement in the organization, but it all comes down to better adaptation to environmental conditions within the previous already existing level of organization. Ideoadaptation includes various change in the color of animals, includes cases of protective and protective coloration. This also includes cases of mimicry associated with a change not only in color but also in the shape of the body. For example, butterflies imitation plants' levels. This is example we speak about last week lecture. Flat body, snape or flounder and other. About change color of animals, it is very excellent example about rabbit. In wild nature, in autumn, in summer and spring, wild rabbit have gray, black color of cover. But when come winter and many snow come to land, they make change his cover to white. In this slide you can to see example of audio adaptation, and in left side you see beak of birds, different all shape, size, long, give to excellent or easy to get food for this different type of birds. Here you can see adaptation too, and here in different pictures you can see fish, lion, owl and frog. They mimicry under swollen river grass which around these animals or fish or birds. And I think the enemy of this type of animals very difficult to see. This is animals. Next, deviation is an evolutionary change in the development of an organ of the middle stage of the formation. It is a common but rare occurrence. Mutation can lead to significant disruption. Recapitulation of observed only up to the middle stage of embryogenesis, and then development following is a new path, leads to the restructuring of the organs. And from this we need to say about recapitulation theory. And you remember about human and chicken embryo, and remember about ontogenesis and philembryogenesis, and what it means for each other of terms. And next part of our lecture we will speak about differences in system of progressive. And in this picture you can see phylum Chordata. Chordates contains invertebrate and vertebrate species. All have bilateral symmetry. All at one point have a knot chord, dorsal nerve cord, pharynx and gill slits. And you can to see all this part in adult and embryo. Next, it is lanceolate. They have fish-like shape. Half or less of their body is buried in the sand. Filter feeders. Have mouths and up to 100 pharyngeal slits along body. And in this place we need to note about development defects. And in picture you see knowing for you examples, and we saw it in our museum. And what is it? Development defects. Unlike fetal embryogenesis, malformations are deviations from the developmental program that reduce the viability of the organism. Disorders of embryogenesis led to the formation of traits in humans that are not normally found in them, but are characteristic of ancestral groups of organisms. And next, the main direction of evolution of integument of chordates. And in this picture you can see vertical section of skin of rocks. What character for this stage of skin? Differentiating into two layers, outer epidermis, inner dermis, and increase in the thickness of the dermis. From a single layer epidermis to a multi-layer. Differentiation of the dermis into two layers, papillary and reticular. The appearance of subcutaneous fat and in the improvement of thermoregulation mechanisms. From unicellular glands to multicellular glands. Differentiation of various derivatives of the skin. And in this picture you are going to see many differences presented. Picture presented derivatives of skin. Many much distance for understand that it same organs or part of organs. Derivatives of the skin are unicellular in cyclostomes, fish, and multicellular in amphibians, mucous glands. Scales are placoid in cartilaginous fish. The epidermis and dermis take part in the development, and bone in bony fish, which developments due to the dermis. Again, it is same, only have different shape, looks, and function. And now in this slide you're going to see skin anomalies. And first it is albinism. This is complete absence of antioxidant pigments. This is picture about albinism down left. In this picture we can to see girls with hair without color. Next, vitiligo. This is pictures of hands and fingers which have dark and light part. Here no pigments in patches. Then in one photo you can to see one family with hypotrichosis, excess hair growth, many hair in eyes, in face. Next, alopecia. Down and right under this family we can to see men with head which no hair, no present hair. Next, hyperkeratosis or thickening of nails. Pachonexia, hair thickening. Polymastia, an increase in number of mammillary glands. This is a picture in middle of the slides. Polytalia, an increase in nipple of cleanness. In this picture you can to see 8 years old Pan Huyanchan from China is called the fish boy by his neighbors. The child suffers from a severe form of ichthyosis, a rare genetic disease that is still incurable. Next, system of organs. It is phylogenesis of the blood circulatory system of chordates. The blood circulatory system has a mesodermal origin. Basis evolution directions: the first, germination and differentiation of the heart from a 2-chamber to a 4-chamber heart. The second, development the second pulmonary blood circulation and a final separation of the venous and arterial blood. The third, transformation of bronchial arteries, arches, and differentiation of vessels branching of the heart. And again, you can see in this slide philembryogenesis or evolutionary tree when we to see many branches. When at first heart was two circulatory lobes, three-chambered heart, then ventricle divided into chambers, then divided to atrium and ventricle. And from left to right side we can to see heart and microcirculatory system of fish, frogs, turtles, lizard, crocodiles, birds and mammals. More and more ready to us it is mammals. And in this slide you can to see description about this system. And the first layers, fish. The heart of the fish has two chambers, an atrium and an ventricle. It is moving slowly on its way to the tissue and needs skeletal muscular action to assist blood to move back to the heart. So the fish relies on physical movement for blood to be recirculated back to the heart. Second layers. Insected are arthropods, and like all arthropods have an open circulatory system. In an open system, a blood-like fluid called the hemolymph flows freely within body cavities, where it makes direct contact with all internal tissue and organs. Again, next layers. The heart of amphibian such as a frog has three chambers, one ventricle and two arteria. In the ventricle, the oxygenated and oxygenated blood are mixed before being pumped out of the heart. And last or fourth layer means the avian heart is more like the mammal's heart with four chambers. This allows for no mixing of oxygenated and deoxygenated blood, thus maximizing the content of oxygen in the blood arriving in the tissue and organs. Now you need to see in this picture and try to find without my notes what happens, why in left and right side of heart differences, and which problem have these patients. And now I give you time for this, and you can to see. Left side have normal heart with separated with many oxygen and little amount of oxygen blood. After use and change oxygen for carboxy, we can to see as a blue, and after lungs with many oxygen, you can to see as right and red color, right side and red color. And we understand that full this lots of blood separated. And I take your attention about between down and up part, when they now in this picture open but can close from door of heart. And the right side you can to see two one situation when differences or separating between left and right part of heart not separated full. It is oval alternative location of ventricular septal defect. This is defect present when heart development and show about before heart was a three-chamber. Then this is a window, oval window, need to close. This is happens when all down chamber is development and became more big. But sometimes in mistake of ontogenesis, this is not close and will baby with not full separated oxygen and carboxy bloods. What wills? It wills the organism, heart, brain and other important organs not take many or enough amount of oxygen, and it will be very bad for development of little organisms. In other side, this is open window show to us fill embryogenesis, stage of fill embryogenesis in short repeat of ontogenesis. And in this slide you can to see other type of defect. This is valve which can close up and down chamber in one side of heart, and this is moment give to come blood from one department to other, and then from heart to aorta, and not back, because it will need close, full close. It is situation will in upper left side of pictures. All three other variants show that it may be stenosis when not three, only two, but I now speak about upper line, normal, and then stenosis when in closed condition that not close full and make little space, and when heart press and give out blood from heart in aorta, some part can return, some part of blood can return. And if it will be every cycle 60 times from one per 1 minute, then every minute, then many periods, it lets that chamber will have many additional blood and will be more and will give many problem of these persons. Down line show other situation. In left side, when we can to say about full open valve, and in this moment blood go away from heart. And in right side, stenosis, and again it not give full open. So when heart have little time for come out this chamber, it is not happens because very small space for heart. If for understanding, I can give example, very simple example. For example, when class finished to study and all students want to go away from classroom, for example, need to open door, and all students will go, and not will any problem. But if this door will open only half, so student will have many problem to go away, and will stay many times in classroom and cannot to come out. No, for example, this is position can discuss when we speak about car and road in town, big town. When we speak about stop hour, when cars cannot to drive in any place. Next part, it is about phylogeneity of respiratory system, and again you can to see line of different type of lungs. First, amphibian salamander lungs are simple air sacs. Amphibian frog air sacs with some branchial branching and compartmentalization. Then reptile compartmentalized expandable air sacs filled by negative pressure. And last mammal, full compartmentalization with gas exchange in small chambers called alveoli. And new slide, and you can to see lungs anomalies, different anomalies when from letters A, not development one lungs, or small and down, B and C consequently of lungs. When bronchial have only one of lungs, it is letters D. When bronchioles present in both lungs, but in one part, lungs 2 and third not have bronchioles. And in letters F, you can to see lungs without bronchus, bronchus without lungs. This is anomalies which happens in philembryogenesis, but if it happens in human, it leads to big problem. Next, about origin and function of the skeleton of animals. Support information in invertebrates representing their constant body shape are very diverse. They are of ecto, ento, and mesodermal origin. In vertebrates, the skeleton is mainly of mesodermal origin. The skeleton in the body of an animal performs a variety of functions, ensuring a constant body shape. Passive part of the musculoskeletal system, protection against mechanical and other influences, hematopoietic function. For example, lancet has a hoard and muscles. Fish have skull, vertebra from two part, and muscles. Amphibians present skull, vertebrae from 3 departments, legs and muscles, and birds and mammals has skulls, vertebrae from 5 departments, thorax, legs and arms, and muscles. And in this picture you can see different variant which was described previously. And here in this slide you can see anomalies and malformations of skeleton in humans. Here the presence of ribs at the lower cervical or at the first lumbar vertebrate, presence of caudal vertebra and spina bifida. Next, he presents of only one auditory ossicle in the tympanic cavity, the column. Heterotopia of the upper limb and girdle. Polydactylia, father and little boy has every hands for 6 fingers. flat feet, club foot, narrow chest, and no chin ridge. It is examples of same anomalies. And last picture show different anomalies of fingers and hands. And it is last slide, and if you have any question, you have to ask me. If you not have any question, so goodbye.